Hello and good morning, everyone. Thank you for being here and your interest in this talk titled Designing Your In Vivo Studies. My name is Somshuvra Bhattacharya, and I'm a technical information scientist here at the Jackson Laboratory. Through this talk, I look to discuss various aspects related to designing and successfully conducting reproducible in vivo mouse studies, sample size planning, and in the importance of communicating methods completely and accurately. Before we go into the meat of this talk, I want to take a few moments to talk about the Jackson Laboratory and who we are. At JAX, we have a mission to discover precise genomic solutions for disease and empower the global biomedical community in the shared quest to improve human health. And we strive to do that by empowering scientific excellence through innovation via discovery, where we have our own research staff with their own private funding that investigate the genetics and biology of human disease, partly by using the mouse or other systems. Through research, we provide scientific services and resources to investigators around the world and education. At the Jackson Laboratory, we are very committed to education outreach. In addition to webinars such as this one you're participating in today, we also sponsor traditional conferences and hands-on workshops, as well as educate students of all ages through courses, internships, and other programs. The Jackson Laboratory is the world's largest repository for mouse models of disease. We are located at 3 different locations throughout the US and also have a location at China. The white dots in this picture denote our presence, Bar Harbor, Maine being our main campus, followed by California, Connecticut, and China. I have certain goals that I want to go over with all of you today, and I'll discuss each one of these goals as we go over them. We'll start off with the main goal, that is designing and successfully conducting reproducible in vivo mouse studies. So to start this topic, I'd like to briefly touch on the 3 R's first in experimental design. The principles of the 3 R's relate to replacement, reduction, and refinement. Replacement includes methods that avoid or replace the use of animals, such as human volunteers or tissue cells, using mathematical or computer models, or established cell lines. So sometimes we have a certain reason to replace, but we are not going to focus too much on the replacement part today, and we'll focus on the reduction and refinement. Reduction includes methods which minimize the number of animals per experiment, and you can achieve that by proper strain selection, that is mouse strain selection, appropriate sample sizing, and accurate and complete reporting of methods so that others can follow. And we'll also touch base on the refinement part, which includes methods that minimize suffering and they improve the welfare of animals. And this refinement can be a broad term that applies to all aspects of animal use, from the housing and husbandry aspects used in scientific procedures, and to avoid stress that is related to handling housing and health issues. Now, since you're here in this webinar, I'm guessing that you have heard about the other R as well, and that is reproducibility. And this is a very big concern in vivo studies. Now, I wanted to discuss the reproducible aspect by discussing repeatability and reproducibility. Now, when you do an experiment in the lab with certain precision, with certain variables, certain factors within your experiment, and you rely on nothing changing, you can definitely achieve getting the same repeatable results over and over again. But what is very important is that biology depends on certain truth and mechanism of how something works and can be tested in different ways, but the concordant results should be the same. In that aspect, it should be also reproducible. And how you can check that is the truth should hold across experiments, laboratories, publications, and even if it relies on parameters changing, yet supporting that one robust truth. So like this cartoon that I put up here, if you look at that, if you are the person who's trying to do that experiment over and over again, it's always a good idea to publish that or communicate that to your peers so that a certain other person can come in and follow exactly what you have performed in another setting, but that the experiments still hold to be reproducible. Now, why is it so relevant? So in the US, we spend around $114 billion annually on life sciences. Out of that 114, around $56 billion or so is spent on preclinical research. Unfortunately, about 50% of that is irreproducible. A high number of peer-reviewed publications involving animals are known to be not reproducible. And perhaps we could say that a high attrition rate in novel therapeutics is a result of that, because we know that when we develop novel therapeutics, preclinical research is a critical aspect in that pipeline. Other than the reproducible parameter from the science perspective, the well-designed animal studies also help you to save time and cost and also support ethical animal use. You'll see a lot more of these aspects when we go into those slides later. So this publication that I put up here, that actually looked at different fields from chemistry, biology, physical sciences, medicine. And if you notice that this trend of irreproducibility is very common in various fields across various domains. And that is also for your own research or for somebody else's research. So this is something that tells us that we need to address this very quickly and very effectively. The other part of reproducibility is publishing that the reproduction attempt. There is also a problem in attempting to publish a reproduction experiment. We all know that in research, it is sometimes quite hard to publish data that is reproduction attempt, or whether it is successful or unsuccessful. So if you look at this data again, published versus failed to publish, that ratio will tell you the story. Now, there will be some gaps in knowledge that probably results in that. And we'll see a quick example here how a gap in knowledge can easily result in irreproducibility. So in this example, let's say two laboratories actually made a knockout of a gene, let's say gene X, and both the models were studied extensively for 10 years. Now I'll open my laser pointer quickly to help you follow. So found to differ, that the results were found to differ in diet-induced obesity and hepatic steatosis. Now, what was not known is what is accounting for those differences. But when the results were out, like lab A and lab B, with the mutation in the FATP1 gene, if you follow what was reported, probably you'll get a cue where this would have happened. The backcross method for lab A was explained that C57 black 6J from JAX was taken 7 times using marker assistance. So they were pretty good in explaining what was the backcross method. Lab B went a little more further with what the original paper had said, and after that, how it was done by the collaborator, explicitly mentioning who did what. The strain maintenance were samely reported by both the labs. But when we talked about when the topic came of refreshing, the lab A did actually mention that it was refreshed 2 times to black 6J with the years as well. Lab B actually missed that data reporting, and the other aspects were more or less reported. Interestingly, the results that was found to differ in diet-induced obesity and hepatic steatosis was actually found to be a result of genetic background. And if this researcher, or I'm not saying that this is bad or good in a way, because this must have been missed, but if you explicitly mention what was the backcross method, what was the refreshing, what was the background, probably this could have been something that could have been avoided. Now, hoping that you understand that these are some of the introductory aspects related to designing a conductor reproducible mouse study, we'll look into more details, starting off with selecting good models and how you can select their controls. So one of the first decisions when designing a mouse experiment is what model will you use? So you have different resources available to make that decision. You start with science, of course, go to your primary literature, go to PubMed, read the papers, and see if somebody has discussed making that model, or if a model exists, essentially. If you're not able to track down that model, you can then go to informatics.jax.org or mouse genome informatics. It is like a one-stop shop for everything mouse, and you can look at if there is a strain in some repository, if it's in live state or is it in a frozen state, and all those kinds of information you will find there. And then you can go to mouse phenome database also. It's a treasure trove of information, and here's the link to that phenome.jax.org, where you can actually read some baseline characteristic data sets and see if something like a body weight or some other basic characteristics have been already studied for that strain or that inbred background, and you can build up on that. So now you have the mouse selected. How do you get those mouse? One option is, of course, go to the different mouse repositories. The Jackson Laboratories happens to be one of the best, one of the biggest mouse repository in the world. You can also go to other repositories like MMRRC and UC Davis, but it all depends on where your strain is. And once again, you can find that through MGI or PubMed. Now, if you find a model in paper or in science and you can't find that mouse available anywhere, so supposedly nobody gave it to a repository, how do you get that? You can always make a new model de novo. You can build up on what you read in the paper and you can make your own model by using various methods. So at JAX, we have our own custom model generation services. Here's the link to that. Please go here and read a little more, or contact us at this email at MyStech with that phone number, and we'll be happy to guide you further how to proceed with that option. Now, regarding the mode of action when it comes to a mouse model, it is important to consider that the relevance of the model is different to what the human disease etiology is. So mice are models. Mice will mimic human disease symptoms, but may not always necessarily follow the same mode of action. One model can only partially reconstruct a human disease, partially reconstruct the human disease. And a great example of this is in these two mice here. The DBDB and the OBOB models are very popular to model certain aspects of type 2 diabetes. However, very few humans with type 2 diabetes have actually a single mutation in the leptin pathway. And for this very reason, it may not make sense to use this model to understand the diabetes etiology in people, but could actually make sense to determine if an experimental drug can manage some of those symptoms of the disease that are shared between mice and humans. So in a way, consider testing multiple disease models and determine whether they're relevant to your hypothesis or therapy, and don't just bank on one model to recreate the whole etiology. Now, model selection can have other aspects that need to be looked into as well. For example, I want to explain this with a very, very good, very commonly used model for type 1 diabetes, that is the Nord-Schild-J mice. These mice are used for studying type 1 diabetes. Note that in this graph over here, let me bring back my laser pointers. If you notice in this graph, you have the males and the females graph with the different colors. With the age and the percent diabetic, you have mice that have variation in that disease onset. Some mice doesn't even become diabetic. And you see there is a variation in the sex as well when it comes to choice of animals. Hence, to incorporate all of these into what can happen, what is possible, it's always a good idea to make sure your sample size is appropriate. If there are changes that is something not known or something that is not predicted, it's always a good idea to take a little extra animals in consideration when planning your experiment. Now, speaking of model sex, as you saw there, it can also influence your experimental design in other ways. Disease prevalence and severity can be different. For example, Rett patients are primarily female, but some mouse mutants have different phenotype depending on looking at males versus females. It could also be that within the literature that there is a skewing that has been noticed towards one sex for many reasons other than biological reasons. Sometimes, for example, male mice are used in neurological sciences research because they are bigger and it's easier to find brain regions or to do surgeries. Sometimes female mice are used in drug studies because they're smaller and you need less drug. So it's very critical to understand that a choice should not be biased on one sex or one's or any other choice other than what exactly the phenotype or the research question is, essentially. Now, NIH also expects that sex be factored into research designs, analysis, and reporting. A strong justification would be required nowadays to propose to study only one sex. So please consider using both the sexes unless you work in a system that doesn't really make sense, like you study ovarian cancer or prostate cancer. Other than that, you should always consider using both the sexes. It would lead you to better understanding of the sex differences, like that variations you saw in that Nord-Schild-J mice results. Both the sexes may be needed to more accurately model the human disease. As I was telling you before, that one model might not be enough to recreate the whole disease. Sometimes you might need to look at both sexes to get you a better, stronger data set. And it also helps you to reduce breeding costs with animal use numbers, because when the mice breed, they're not going to preferentially breed male or female. If you plan to use both the mice, you're definitely going to save a lot on the breeding costs. The other aspect is considering the phenotype. So genetic models do not equate to disease uniformity. You need to understand that one model doesn't always going to give you the same type of phenotype over and over again. Mice are living organisms, they're not machines. It just has to fall within a certain standard deviation. And if it falls within that standard deviation, you should accommodate those factors when choosing that model. things like age, sex, genetic background, environmental factors can all affect the phenotype. And sometimes the mice might not even show that phenotype that you are planning to see. So it's always a good idea to keep these things in mind when you're planning your mice experiments. And then selecting your controls. So there could be a multiple ways you can selecting controls. Today we'll talk about selecting the controls based on genetic background. A very common example that comes to us a lot is how C57 Black 6J and C57 Black 6NG are different from each other based on their genetic makeup. So if the researcher doesn't distinctively mention in the publication or in the report what kind of mice, what substrain was used, it can easily get very confusing. Now, these two are two different substrains, and it's very critical for you to understand what could be the differences that would come with choosing the wrong mice. And if you are not careful and you start breeding, for example, very soon your progeny will be having one or the other and show those phenotypes, which is not expected. Now I'll show you an example here of why knowing that genetic background and substrain is really important. Genetic background is known to influence phenotype, and this is an example coming from Mouse Phenome Database, where they took an FEV strain. Let me bring back my pointer. This FEV strain that was a model for mammary tumors. And what they did was they took transgenic males and bred them to different female inbred backgrounds, all of them here. Then they measured the tumor growth after in the offspring. And what you see here that depends on what background it was crossed with, you have variations in when the tumors showed up. Now, it was the same source, but when they're crossing, actually the different backgrounds are accounting for this. And how this is important is, for example, you're planning a drug study with that tumor and trying to make a good, a design for a control mice versus experimental mice. If you're not careful and this mice is crossed to this strain, you see it takes almost 80 to 90 days for the tumors to show up. Whereas for this on the right-hand side, you see it will be almost like 40 days or so around that the tumor shows up. So that's a lot of difference. So if you use the same, if you're not careful and if you use the same thing that has been showed here, easily you can have different systems showing tumors at different times. That is beyond your control, essentially, to predict when those tumors will show. Hence, it's very essential for you to understand the background and what influence it has on the phenotype. And as I was mentioning before, substrain divergence is another aspect you need to consider, and that could be happening if you are not careful, or it could also happen like substrains can generate by themselves over 20 plus breeding generations. Phenotypic differences between the parent and colony can be discovered. If you look at the nomenclature, sometimes that is a very good lead for you to know. We actually have a dedicated webinar for mouse nomenclature, and I'll be happy to share with you all that information if you need. But you can actually look at the name over here, like for example, the parent strain, the substrain designation, and which laboratory is maintaining that strain to know exactly what substrain they are, so that you don't mix any one of these with another strain. So genetic drift, as I was mentioning, can easily contribute to data variability. A well-known example over here is the substrain divergence can be seen through the C3H mice. So C3H and its related substrain was initially maintained by Dr. Walter Huston, who worked at JAX for 2 years during the 1930s before moving to a newly established National Cancer Institute in Bethesda in 1940. In 1952, he actually sent some mice to Henry Odson, also at JAX, and both strains were ultimately transferred to our production campus. Now, what was different is in the late 1960s, investigators discovered that the C3HEJ were becoming resistant or were showing signs of resistance to the effects of lipopolysaccharide, LPS, whereas most of the other inbred strains, including all other C3H substrains, remain sensitive to LPS. The mutation responsible for this phenotype was ultimately mapped to the TLR4 gene, which actually plays a key role in pathogen recognition and activation of the innate immune system. But by the time this mutation was identified, it had already become fixed in the C3HEJ substrain, having likely arisen somewhere between 1958 and 65. So now if your experiments involve an endotoxin resistance, you should always be aware of which substrain is resistant and which is not in order to do the right experiment. Now you can avoid genetic drift or any kind of unnecessary drift that happens in your colony through genetic stability. One way is to know the history and the genetic background and substrain of your mouse. Having good pedigree records, knowing the genetic quality is definitely critical when you're taking mice from a collaborator. But one way is you can go to mouse repositories like the Jackson laboratory. All repositories should maintain genetic quality. Purchasing cohorts, new breeder pairs, or cryo-recovering mice is a relatively small investment in the long run. You can also verify the genetic background with tools such as genome scanning or GigaMoga panels. So we at Jackson Laboratory, we actually take this a step further. We actually do we do this because we want to make sure that the phenotypic data that you collect from researchers such as I showed you, the mouse genome database, is not only comprehensive but also consistent over time. In order for this to occur, we need to ensure that the genome of our mice is stable over time. So we have a patented genetic stability program, and through our patented program, approximately every 5 generations, our foundation stocks of some of our most popular strains are reinfused from cryopreserved embryos, thereby limiting genetic drift. So the Black 6J mice or the FEPNG mice that you receive from JAX today, and the data that you derive from them will be as similar as possible to the mice you receive in 5 to 10 years. And we do this genetic quality maintenance by various steps, including using the help of highly skilled animal caretakers who intensively go through training programs, who have been subjected to various lab science, lab animal science and genetic courses. We use rigorous colony management protocols, and we also do systematic screens for variant genotypes and phenotypes that include quality control SNPs, allele-specific genotyping assays, and of course look for coat color changes and strain-specific phenotypic validations. Now, it doesn't mean that you can do that at your institution. You can use good colony management practices to minimize this impact on your phenotype. One thing to avoid would be to avoid selecting mild or strong phenotypes just because it basically put bias in your mice choice. Don't select a big mice or just because of breeding purposes. Always remember, phenotype for your mice is more important than anything else. Maintain a defined breeding rotation and a mating scheme. Keep good records, keep pedigree records. Phenotypes can appear sometimes after breeding, so keeping a note of them and keeping a regular observance on those mice is very critical. Refresh the genetic background regularly and cryopreserve some of the unique strains. And I want to talk a little bit more about like natural calamities or disasters like what we are going through right now, like COVID or the hurricanes that we see nowadays a lot. Sometimes it's just not possible to, you do have to leave your facility, perhaps personal changes happen, or sometimes you just have a strain that is not going to be researched upon. And it's just a waste of resource to keep that strain alive. So it's always a good idea to cryopreserve those strains, those unique strains, so that you can rederive later and start your experiments all over again. The Jackson Laboratory provides various options for cryopreserving, and once again, I'd be happy to help you, or my teammates would be happy to help you walk through them. Please contact us at the contact details that will be provided later. And you can also purchase cohorts from a trusted vendor. Just buy new mice, and that will help you to minimize your genetic drift or chances of genetic drift. Now, hoping that you now know that how important it is for choosing a good mouse, the next part of it will be choosing the assays that you would do. The assay selection will depend on the in vivo model. Knowing that your mouse again comes into play when choosing an assay, because, for example, an inherent strain characteristic can easily dilute your assay. For example, let's say a mouse suffers from hearing loss, and in your experiment, you plan to use startling response as one of the parameters for your experiment. If you're not aware that the strain that you have chosen is known to have hearing loss, probably they will not respond to that starting response. So it's always good to know what the strain characteristic or the inherent characteristic is. Looking at repeated measures like locomotor assays, sometimes mice get used to it if you make them run on a rotorod. And if you use the same mice over and over again for your experiments, probably they would adapt to that rotorod and they would know what to do. So it's always good to change those, rotate those mice in your experiment and see if that phenotype or that feature is being exhibited truly by the mice. And select many complementary assays. So as I was saying about the mouse model, it same holds good for the assay. One assay might not be enough or adequate for you to show the phenotype that you're trying to see. Sometimes you might have to use multiple assays to recreate more aspects of the disease or more aspects of your experiment that you look to study. As I said before, assay may not fully model a process. use several complementary assays to test your hypothesis. And very important aspect is understanding the pros and cons of each one of them so that the second assay or the complementary assay would be able to overcome that. And sometimes a mouse assay may not essentially recreate what is measured in the patients. So develop new assays which you think will better recapitulate those clinical measures and give you a better quantifiable data. And it's absolutely fine to do, mix and match or cross between two assays if you think that that is going to answer the question better. Now, handling stress can affect experimental readout as well. Implement mindful handling practices. Mice would be very sensitive to handling practices and what kind of husbandry practices is being practiced. Plan for consistency in the experimenter, keep notes. Sometimes it's not possible or feasible to keep the same experimenter, but if you keep good notes, then the next experimenter can follow those notes and do what exactly was done before. Reduce handling to a minimum, and remember that all of us have one goal, and that is to promote scientific discovery and good welfare. And in all those aspects, if you follow the right measures, probably this will be one of those aspects. Remember, the refined part that we were talking in the beginning could be taken care of better than others. Now we'll move to sample size planning for our goal 2, and how this, when you design an assay, when you have a model, how those sample size can be adjusted for a better in vivo study design. Mice being biological reagents can show differential phenotype. There will be a natural variation in their phenotype onset. What you see in vitro sometimes may not match what you see in vivo. If you use human donor cells, for example, in your experiment, that can also contribute to variability. And based on your assay, you are actually trying to see whether you are having too few samples or too many samples. If you have too few, experiment cannot provide feasible answers, of course. But if you now have too many, you're actually wasting resources for absolutely minimum to no gain. And here comes the power of statistics, how you can use them to your advantage. Let's start with p-value. It helps you to understand the probability of measuring a false positive. It will help you to figure out if there is a difference between the two groups, but it won't essentially give you an indication of how large that difference is. A statistically significant p less than 0.05 can be obtained in various ways. And now you must all be aware that a p-value is very, very field dependent. Sometimes a field can have a different p-value as acceptance in what they're studying, and the other field might have a different p-value what is accepted. This value will help us determine if there is a difference between, once again, the two groups, and does not essentially tell you the large difference or if there is any quantify that difference for you unless you use some other parameters. You can adjust the p-value by tweaking the sample size or the effect size or doing the other way. But you can make this even more powerful if you add some more features like the effect size. Now, how large that difference is between the two groups, if your question was, can I quantify that? Yes, you can. You can use the effect size. This tells us how well our intervention worked. An acceptable effect size can vary once again between disciplines, but generally speaking, an effect size of 80% is acceptable. But some fields might also consider an effect size of 20% to be acceptable. It's always a good idea to read literature and talk to your peers or see what's out there to what is accepted in your field. But the confidence interval, when you report it with the effect size, it will probably tell you the probability of a finding of being reproducible. Now, there are other factors that you can rope in your studies as well. For example, pilot studies and power analysis. They can all help you to determine your sample size and maintain a better sample size planning. Power will be the probability of finding the true difference between the groups. Acceptable power is, once again, like the other two factors, can be very dependent on the discipline that you are in. It shares a strong relationship with p-value, effect size, and sample size. So all of them combined gives you tremendous power as compared to each one of them separate. The pilot experiment is always a recommendation that we give, and we discuss this a lot with a lot of researchers, is whenever you have a doubt, you're doing a new experiment, you do not know the phenotypes in your in vivo experiment, start with a small number of samples, start with small n, choose an assay, collect that data, and calculate your p-value, effect size, and power, and see if your pilot is underpowered, or you can increase that power by either you can increase your sample size, or you can keep the same sample size, but you can choose new assays that show better phenotypical effects. Experimental planning and statistical analysis can go always and needs to go always hand in hand. Sometimes people from life sciences might not be as experienced in biostatistics as many other fields might be. So always, if you find a biostatistician, a lot of institutes already have biostatisticians in their own institute. Use their help to design your experiments. They will be very critical to help you design those sample size parameters. Perform pilot studies with those planned parameters that you plan to use in your study to calculate a power and estimate your appropriate sample size. Randomize your study, remove the bias if you can, and blind the study if possible so that it doesn't creep into your data and dilute the results. And if you still can't do, if you feel that there is a lack of reproducibility, things are not working out, please report the p-value and the effect size in the statistical analysis you've used when you publish those findings so that the next researcher after you can at least build up on where you stopped. And finally, that brings us to the last goal of this study and how important is communication and importance of that in research, in vivo research, essentially. So I brought this paper up here. This paper talked about survey of 271 animal studies with 72 on mice. And a lot of those studies actually had uncertainties in strain, sex, age, weight, randomization, and sample size or statistical parameters used. This is alarming because reproducibility relies on accurate and complete reporting of methods. And if that report is true in what it has found, that means a lot of researchers are probably not reporting the data or reporting the statistics as they should. NIH does encourage greater scientific rigor and transparency in experimental results. So reporting your in vivo experiments needs to have to go through some rigorous planning. And if it helps, you can use the ARRIVE guidelines as a tool to improve communication and reproducibility. Now, how can you use the ARRIVE guidelines? So the ARRIVE guidelines were developed by the National Center for the Replacement, Refinement, and Reduction of Animals in Research. So if you look at it from a research point of view, it looks like a research poster essentially, with the title, abstract, introduction, what we do when we write a research poster. You can go to this link over here that talks about all the 20 factors and what needs to be discussed there. These are like guidelines for a researcher to report when they're writing a paper. But the ARRIVE guidelines were actually tweaked a little bit recently to make ARRIVE guidelines 2.0. Understanding the fact that sometimes those 20 factors cannot be mentioned in the papers as clearly as it should have been, but then the ARRIVE guidelines was tweaked to incorporate the essential 10, and this is a checklist of the 10 things that absolutely should be reported. This includes study design of what your design was, what was the scheme that you probably were using, the sample size, inclusion and exclusion criteria, what you thought would not be important, randomization, blinding, outcomes, methods, results, experimental details. These will all be very critical for the next researcher and your community or your research community to follow based on your findings, essentially, if you follow these ARRIVE guidelines. Some journals have already started endorsing these ARRIVE guidelines, and I'd like to point out Nature and the Nature group of publications, they more or less endorse the animal research ARRIVE guidelines when reporting in vivo experiments. Nature is also including actually a section called the reporting summary, where researchers must report on whether or not they reported important experimental details. Think about it as a checklist of things that, hey, I got this one done, I got that one done, so that you know that everything was taken care of. It also helps you as when you're drafting your paper to know that you didn't miss anything. And after all of this, what if you still can't reproduce experimental results? It's quite possible. Then you need to consider that other factors could be playing a role in your findings beyond what you have tried to recreate over and over again. Consider that the possibility that the research that you started off by reading about or the original findings are probably not reproducible in the first place. And after all of this, do you think that this results is all waste? Absolutely not. Please publish those negative or alternate findings. Sometimes negative data can help you achieve great wonders, because the next researcher knows what you did, what was wrong, or if there was indeed a wrong, it's research. The next researcher just picks up from where you left, and that's how the ball rolls, right? And one great example of after all of this, it doesn't work is this example here on the right, where a lab was located actually in Seattle. This is a publication. Please feel free to go and read the whole paper. But in a gist, this lab was located in Seattle, and leptin administration in that lab in Seattle was found to show decreased food intake in the animals, along with versus saline, as you can see here, with the solid bars being leptin and the saline being the white bars. Now, the researcher's name was Woods, who's the author also. So Woods moved his laboratory from University of Cincinnati, along with the technician who had personally conducted those leptin experiments, to Seattle, and everything was done the same way. And once they moved to Cincinnati, the same technician tried to replicate the leptin experiments in these new surroundings. However, despite using the same source of leptin, same supplier and strain of the animals, it was found that if you see the results, that it was just the opposite results of what they saw based on leptin administration and food intake. This was repeated over and over again and shown the same results. And according to Woods, at some inexplicable point, after many negative experiments, leptin was once again found to reduce food intake in the laboratory. And it has been relatively reliable anorectic agent ever since. So it was not explained why this happened, what was there. But one good thing to know is, so if you, this is some factor that we never even discussed today, right? Or you might not even thought about that. Geographic location could also influence. You might even need to consider that the original findings were not reproducible. Many things could all factor in either singly or as a combined factor to affect what you're seeing in there. So if you're moving lab or you're moving places, you're moving technicians, repeat those experiments, look at all the possibilities to see what could be driving those differences, if they are, and is this consistent over long periods of time in different conditions. So with that, we would like to finish today's talk. Summarizing what we just discussed: reproducible in vivo research studies depends on a thorough understanding of what mouse model you have chosen. Biological factors, colony management, animal husbandry, experimental behavior can all influence consistency, and just good communication, good statistical, well-powered studies can all help you achieve a good in vivo study. When you look at this graph on the right that I made, if you think about your in vivo study as the ball in the blue in the middle, it's a very good tightly knit procedure where you have to make sure all of them are in the same tandem with each other to achieve superior results in your in vivo studies. And with that, I would like to finish today's talk. This is our contact address, mystech@jax.org. We are a group of very friendly and very, very helpful technical information scientists at JAX. We strive to help you with all your mice research, with all your needs, all your questions. Just give us a call at this number or email us your questions, and one of us will definitely get on board and try to help you from there. And I also wanted to spend a little time talking what we do. JAX is available to support your in vivo studies in many therapeutic discovery areas with model platforms, services, including target validation, in vivo pharmacology, safety and toxicity, and many more. We can help you in whatever step of your mouse experiment you are in right now. Once again, please contact us to let us know how we can help you and achieve your research goals, and we'll definitely have a conversation going.